Um, Today I'm going to talk to you about polymeric microneedles, microneedles made from different polymeric formulations, and how we can use those for transdermal and intradermal drug delivery. I'll also mention some of the opportunities I believe that we have in this area, and also some of the challenges and how we might overcome those. And I'm going to concentrate particularly in long-acting drug delivery applications of microneedles. So, if we can go then to talk about the overview, I begin to talk about long-acting drug delivery, and then I'll talk about some of the issues with current systems for long-acting delivery. And then I'll move on to talk about how microneedle systems could potentially be useful to overcome some of these challenges. And I concentrate in particular on microneedles made from hydrogel forming polymers and how we can alter their design and do that in a flexible way to allow delivery of both hydrophilic and hydrophobic compounds. And at the very end, I'll talk about the translational development. How do we actually go from laboratory innovation to a product that may actually be able to help patients? So long-acting drug delivery systems, as the name suggests, involve slow absorption of a drug, and the role of long-acting drug delivery systems is clinically and medically beneficial for the treatment of chronic diseases and chronic pain. So very often, if a person has a chronic disease, they will have to take one or even multiple tablets every day. And this can be a reminder that they are suffering from a long-acting condition, and this can cause a number of psychological issues, which lead to poor adherence with the treatment. And this can lead to relapse, or it could lead to development of resistance if it is an infectious diseases. So one way of making things more convenient for patients and preventing poor adherence is to use a long-acting delivery system. Now, that can include an injectable, so that could be an oily injection with a hydrophobic version of the drug that would be absorbed slowly over weeks or months. We'd use an implant that is generally a solid or that forms a solid upon injection. You could use a transdermal patch that could be worn for up to a week, depending on the quality of the adhesive. And then we can have things like intravaginal rings and compounds like monoclonal antibodies that are inherently long-acting because of their long circulating time in the body. And a major driver, as I mentioned, is to reduce this burden of compliance on patients as compared to frequent dosing associated with traditional pills. It may also be an advantage to avoid the first pass metabolism there for improve efficiency of delivery and reducing side effects due to the production of toxic metabolites during that first pass through the liver. But injections and implants are invasive and they require intervention from a skilled healthcare professional. We can do that in developed countries, but in developing countries where we have a real lack of skilled healthcare professionals, then we have a problem. And we also have an issue with disposal of needles and used medical sharps in developing countries. And this can act as a barrier to adoption of long-acting therapeutics, and then we end up with poor compliance with treatment. Even in developed countries, up to 20% of people have severe needle phobia, and they won't even get regular vaccines as a result of that. So I think that if we want to make long-acting delivery as successful as possible and reach as many patients right across the world as possible, then we need an alternative. And this is where microneedles come in. So microneedles, as the name suggests, are arrays of tiny needles on a solid support. What they all have in common is that they're less than 1 millimeter in height, so that whenever they go into the skin, and the rotating image you see here is an optical coherence tomography scan of somebody's skin where microneedles have been inserted. We don't reach pain receptors, we don't reach blood vessels, so we don't cause pain or bleeding, but we do form water-filled holes in the person's skin. And we can make microneedles from polymer, from glass, from silicon, from metal. We can make them in all sorts of different shapes. But typically microneedles have a small patch size. And what this means is that the dose we can administer tends to be quite low. And so almost all of the papers you'll read on microneedles are concentrating on vaccine delivery. But I see microneedles as having much greater utility than that. And I think that if we can consider higher loadings of microneedle systems, so moving away from traditional coated microneedles where only a small amount can be coated on their tiny surfaces, to polymeric microneedles that might contain the drug or could be associated with a drug reservoir. And we think beyond a 1 square centimeter patch, then all of a sudden then the potential for long-acting drug delivery where we might have to give tens or even hundreds of milligrams for a long-acting dose, that becomes possible. So we started off with dissolving microneedles, and we just made these from a combination of polyvinyl pyrrolidone and polyvinyl alcohol. And so we took those polymer gels, and into those gels we mixed poorly soluble particles of a number of different HIV compounds. So these are typically used in an injection of nanocrystal forms of the drugs injected intramuscularly at once a month or once every 2 months. And what we did here was to follow the plasma levels in rats for microneedle systems. So first of all, you can see with rilpivirine, over a 56-day period after a single application of these dissolving microneedle systems, that we were still above therapeutic plasma levels of rilpivirine even after 56 days in the rat. When we looked at etravirine, we looked at two powder forms of the drug. So we ground it up into stabilized nanocrystals, or we just used the powder direct from the manufacturer, mixed it into our gel, cast it into the mold, and then we had the microneedles dried and we then applied them to the rat. Again, the microneedles were only applied for 24 hours, long enough for them to dissolve in the skin, and then they were removed. And then we followed the plasma levels, again, in this case, over around about a month. And we found that we had therapeutic plasma levels of etravirine for that entire time. We then moved on to cabotegravir, and we compared the microneedles to an injection of the proprietary product intradermally or intramuscularly. And then we looked at different forms of the microneedles. So we used either the free acid form, the long-acting injectable form directly from the injection product, or we used the more water-soluble sodium salt. And what we found was that the intramuscular and intradermal injections both give higher plasma levels over 28 days, but that the microneedles still maintained therapeutic plasma levels for the entire period. And after a month, the more soluble water-soluble salt had equivalency with the injections. When we move to bictegravir, which again is another poorly soluble form of a HIV compound, then what we saw was that the microneedles, whether we use the nano suspension or just the micro suspension, which was the powder straight out of the bottle to make our microneedles, we basically had equivalency with the intramuscular injection for a month. Allometric scaling of long-acting delivery systems is extremely difficult. So here, flip-flop pharmacokinetics are operating. It's not the elimination phase that's important for overall pharmacokinetic exposure. It's the slow absorption phase. So predicting human pharmacokinetics from a rat is not so easy. But if you engage in some physiologically based pharmacokinetic modeling and you consider then the deposition data in the rats, then what we can say is that for around about a week after application of a patch of around about 30 square centimeters, we would be able to maintain therapeutic levels in a person. So instead of having an injection every month or every 2 months, the person would be given a box of patches, they would apply them to their own skin, and the microneedles would dissolve in the skin and deposit the long-acting form of the drug for sustained delivery over a week. And then the next week, the person would apply another patch. I actually think that a person, from my experience in community pharmacy, would remember to apply a patch every week rather than every month. And obviously the microneedles dissolve in the skin, so there's nothing to dispose of at the end. Another technology that we can use to obtain sustained or long-acting drug delivery is hydrogel forming microneedles. So this is a technology developed here in Queens that is unique to our group. Here we use cross-linked hydrophilic polymers that swell in the skin. The microneedles themselves contain no drug, but the drug reservoir sits on the top of the microneedle array. So the microneedles swell, and then the drug will be hydrated by skin that comes up from the interstitial fluid, and then the drug will move down its concentration gradient at a rate that we can control by the cross-linked density of the polymer, the chemical nature of the polymer, and the nature of the drug-containing layer. And this means we have potential for higher doses and potential for sustained delivery, but only while the patch remains, obviously. Importantly, these microneedles are removed intact from skin and they don't deposit any polymer in the person's skin. And here we have some instances of water-soluble drug delivery. So we looked at a series of different antibiotics, tetracycline, levofloxacin, metronidazole, but we also looked at bovine serum albumin and the Alzheimer's drug donepezil. And what we found here was that basically, while the patch was in place, we continued to deliver drug, and in many cases, the plasma levels were still rising at the end of the 24 hours. And what that suggests is that there's still more drug in either the reservoir or in the swollen polymer microneedles. And that means that if we continue to leave the patch in place, then we could continue to deliver drug and still remove the microneedles intact at the end without depositing polymer in the skin. So we're typically using either Gantrez, which is polymethylvinyl ether maleic acid, cross-linked by esterification with polyethylene glycols of different molecular weights, or we're using polyvinyl alcohol, cross-linked by citric acid by esterification and strengthened by polyvinyl pyrrolidone as the hydrogels here. And here we have the Gantrez system employed in mini pigs. And what we found was that we were able to maintain constant plasma levels in the mini pig for 5 days with zanamivir, which is a potent antiviral drug. And the patch was completely removed intact at the end of the 5 days. So that means we're not depositing any bulk polymer in the skin of the animal. And so there's a lot of potential here for sustained drug delivery while being able to remove the patch at any time if there was to be any side effects and not depositing polymer in the skin. And we also are not limited by what we can load into the microneedles like we are with dissolving microneedles. Here we have a situation where the drug is in the drug-containing layer that sits on top of the microneedles, and so we're not actually limited by what we can load into the needles. We wanted to see if this was feasible with poorly soluble drugs as well, though, because zanamivir hydrochloride is very water soluble. So we decided to look at atorvastatin, which is a poorly soluble drug used in modulation of lipid levels in hyperlipidemia to prevent cardiovascular disease. So here we used a special reservoir based on a hot melt of polyethylene glycols, low molecular weight and high molecular weight, to form a waxy solid reservoir. And the idea here is that polyethylene glycol will form a co-solvent in situ with the water from the interstitial fluid and will keep atorvastatin in solution in the swollen hydrogel microneedles as it diffuses down through the hydrogel and partitions into the viable skin. What we think happens then is whenever the polyethylene glycol disperses, that the atorvastatin drops out of solution and forms individual nano or micro scale depots in the skin that will then slowly dissolve and be absorbed. And that's exactly what was borne out in the rat study, where we found obviously oral administration gave a high peak plasma level, but that the microneedles allowed maintenance of human therapeutic levels for 14 days after a single application over 24 hours of microneedles. So potentially we have a patch that could be worn for 24 hours, but could deliver enough drug that would sustain the administration of atorvastatin for a full 2 weeks. And this could reduce the pill burden on patients who are taking this chronic everyday medication. We looked at this with the HIV drug cabotegravir, but instead we used the cyclodextrin reservoir. So as you know, cyclodextrins are C-shaped molecules with a hydrophobic cavity that a drug substance can fit inside. They have a hydrophilic outer surface. So essentially they act as a cloaking device for poorly soluble drugs. So we made a lyophilized wafer containing the cyclodextrin cabotegravir complex, and then we applied that to the top of hydrogel forming microneedles and followed plasma levels over 28 days after a 24-hour microneedle application. So obviously the drug was diffusing through the microneedles, partitioning into the viable skin layers, and then we were having dissociation of the complex. Drug dropped out of solution and formed a depot in the skin. And we had pharmacokinetics that were quite comparable to the intramuscular injection of the proprietary product. And that means then we have a situation where the microneedles could be used to deliver this hydrophobic drug to form a depot in the skin, but the microneedles are removed intact, and we have complete control over the drug loading, and that means that we have some ability to reduce patch size. So I think that this is quite encouraging, but we want to make sure that hydrogel forming microneedles are safe. So we looked for microbial content using the standard British pharmacopoeia test, and we couldn't find any microorganisms in the system. We deliberately spiked the gels with gram-positive, gram-negative, and fungi, and there was no survivors from the production process. And actually, the surface of the hydrogel forming microneedles, as you would imagine, with a hydrogel exhibited anti-adherent properties, and the endotoxin levels were below all pharmacopoeia limits. Obviously, microneedles penetrate the skin, and this creates pores in the stratum corneum right through down to the dermis. We want to make sure that we're not delivering commensal bacteria in significant amounts into the viable skin layers. So what we did was we grew Pseudomonas aeruginosa, Candida albicans, and Staphylococcus aureus in radio-labeled thymidine, and then we seeded a lawn of those on top of some piglet skin. We then punctured the skin with our microneedles, and what we found was that there was significantly lower permeation of microorganisms through the holes in the skin compared to if we punctured the skin with a hypodermic syringe that would be used to deliver insulin. And we know that diabetics aren't continuously getting skin or systemic infection despite not cleansing their skin prior to testing or insulin injection. And as a result of that, we think that microneedles are going to be very safe from a microbiological point of view. And actually, we've just been part of a big publication from PATH's Centre of Excellence for Microarray Patches that suggests that the likelihood of any clinically significant infection arising from microneedles is minimal. Obviously, regulators may insist on an absolutely sterile product. So we took the hydrogel forming microneedles made from Gantrez and we irradiated them with 60 kilograys, which is pretty much the highest dose you would use for irradiation. And we found that their swelling, their physical properties, and the permeation of both small and large molecules was not actually affected significantly. So the microneedles can be manufactured in a normal environment in a GMP factory, and then gamma sterilized and then brought into an aseptic environment to have the drug containing layer added and be packaged. But obviously, if you're delivering drugs with microneedles, you might need to apply the microneedles on a regular basis. So what we did was we got a number of human volunteers. We applied microneedles to their skin every day for a week. And then we looked at the skin barrier function, we looked at skin appearance and a number of biomarkers. What we found was, obviously, when the microneedles are removed, you see a little bit of erythema, but it always resolves overnight. And the transepidermal water loss immediately after removal is elevated because we've created holes in the skin, but it returns to baseline levels the next day. And then if we look at immunoglobulin E for allergy, C-reactive protein for infection, immunoglobulin G for an immune response, and tumor necrosis alpha for an inflammatory response, we found that at the end of the week, there were no elevated levels of any of those biomarkers. So it seems that repeat application of hydrogel forming microneedles is going to be safe. I did mention about the patch size for long-acting delivery in particular, but also for a range of other drugs having to be greater than 1 square centimeter. So what we did was we made a large patch that was made up of 16 individual microneedle arrays. So the overall patch size was 16 square centimeters. And we got people to apply that to their skin using a pressure-responsive film that basically would change color when the correct pressure had been applied. So if a little corner of the big patch hadn't been applied, person will see no discoloration. So if they need to press onto that again, they'll know that the microneedles have been inserted. So we used optical coherence tomography, which is the visual analog of ultrasound imaging, to basically work out what the insertion depth was of microneedles on an individual 1 square centimeter patch, and then at various different points on the multiple patch. And then we looked at the pore width and we couldn't find any significant differences. So people can apply large patches. They don't need an applicator device. They just need some visual feedback that each area of the patch had been properly applied. And this is the sort of setup we think that would be required for actually making a final product. And we're working with a number of different companies to develop this technology. And we think that an integrated all-in-one patch can be manufactured relatively easily. So I think hydrogel forming microneedles and polymeric microneedles in general have great potential for delivery of a wide range of molecules, including hydrophilic and hydrophobic, large and small molecules. High dose administration is possible, as is long acting delivery, and we can be flexible in formulation to suit individual needs, whether we're delivering a hydrophilic or a hydrophobic drug. We're working with a range of big pharma companies as well as smaller companies. And we have a major EPSRC program ongoing to de-risk the platform. So basically show that the platforms themselves are safe and can be scaled up in terms of manufacture. And this will allow us to develop a bridging program that can be applicable to any drug to be delivered. There has been considerable progress made on scalable manufacturing in industrial setting, and I just want to mention LTS Lomond, who we work with very closely. They are now working with the vaccine company, Micron Biomedical, to scale up manufacture of measles and rubella vaccination for a phase 3 clinical trial. I actually think that could be the first microneedle product. And once that reaches the market, then I think we'll see a range of other non-vaccine products coming along. And we have 2 clinical trials of our own technology scheduled in the second half of this year, both for systemic administration and also for local administration. So I would like to think that after several false stones and quite a long period, that we will now finally have a realistic chance of seeing microneedles commercialized and the undoubted patient benefit that will flow from that. So I'd just like to thank the members of our group who have produced the data I've presented today, the sponsors of our work, and also to thank you again for your attention. Thank you.
Thank you very much, Ryan, for this very